So this next video is basically a further elaboration on the component and the function of each histidine kinase and also the response regulator. So we'll start off with the histidine kinase, and as per in the first video, so histidine kinase mostly are periplasmic membrane receptors because they have to be there to receive the stimulus from outside environment. So as you have seen in the first video, okay, so it has a homodimer structure, so it functions as homodimers, which means that autophosphorylation, of course, okay, is a biomolecular event, okay, involving lots of biomolecular components, protein, DNA, metabolites, and so forth. So hence, okay, the domains, okay, are periplasmic and N-terminal binding domain. So apart from that, there is also transmembrane domain, okay, the one here, okay, that will receive, okay, the signal, and it also has a linker domain, okay, and histidine containing phosphotransfer domain, which is over here. So it also has a C-terminal kinase core, and okay, some of this, okay, for example, okay, the Ki and the NTRB, okay, are soluble cytoplasmic histidine kinase, but not all of them are as such. So the histidine kinase, okay, this is more or less, okay, it has a catalytic core, okay, with 350 amino acids in length approximately. So we know that the structure is a dimer, so it has ATP and ADP binding and phosphotransfer domain plus phosphatase activity, which are found in some but not all TCS. So these are the two types. Now, the histidine containing phosphotransfer domain, okay, is about 120 amino acid in length, and it has a histidine residue, but no kinase or phosphatase activity. And the last one, the last two, okay, we have a sensing domain, okay, which is basically at the N-terminal, okay, and it senses the external stimuli, okay, and of course, okay, it is commonly referred to as periplasmic receptor, but not always, okay, because it also depends on the condition of where the system is. So in many cases, okay, the ligand, okay, or the stimulus is unknown, okay, and of course, okay, the sequences in the sensing domain has little to no sequence similarity to any other sequences available throughout the prokaryote. So the last one we have is the transmembrane and linker domain. Okay, so these two domains are poorly understood, okay, because it is not known what are their specific function. But okay, it has been known as much, okay, that they are critical for propagation of signal from the periplasmic binding domain to the kinase core, kind of like something, a bridge, okay, that connects them. So we move on to the second element, okay, the response regulator, because without which it wouldn't complete, okay, the two-component system. So it is it comprised of N-terminal receiver or regulatory domain, as you can see over here, okay, which an ASP. So it also has a C-terminal effector domain, okay, the one that will give, okay, the output. So it is a DNA binding transcriptional regulator, okay, with enzymatic activity, okay, for example, KB or reg-A, okay, and it has a protein interaction or gives out protein interaction. So all in all, okay, it catalyzes the transfer of phosphoryl group from the phosphohistidine kinase to conserve aspartic acid, okay, here, okay, and okay, the phosphorylation that occurs result in conformational change of the response regulator. So apart from that, many of the response regulator also catalyze autodephosphorylation, which is something that is quite common or automatic. So this is more or less, I guess, some sort or some mechanism or kind of like types, okay, of organization of TCS. So as you can see, okay, three of them are given from E. coli, okay, one is from Bacillus subtilis. So for osmoregulation, okay, in the osmosis, okay, or for homeostasis, okay, so we have, okay, different kinds of reaction that the TCS gives out, but it is always the histidine kinase over here and the response regulator. Now, as you can see, okay, over here, the ones that change or the one that differ quite a lot, okay, is basically the response regulator, because it is the one that receives the signal from the histidine kinase. So, for example, for the osmoregulation, as you can see, this is the classic case of the basic case of the two-component system, whereby, okay, the histidine kinase, okay, receive the stimulus from the environment, basically it autophosphorylate and then gives out the signal, or basically transfer the phosphorylate to the aspartic side, aspartic acid side of the response regulator, and this one will automatically dephosphorylate to give out the appropriate signal to turn on the gene or to turn off the gene, and so forth. In case of adductic redox regulation, what happens is that so your response regulator basically receives the signal from histidine kinase. So the phosphorylated signal is basically transferred to another histidine. Now this is what happens. So it does not give out the signal as yet, but it will basically be phosphorylated again to another aspartic acid site and this time another, of course, an example, because this is in Gola, another gene will be transcribed or expressed. And in the third example on the chemotaxis, so basically what happens is that this is because it has two kinds of reaction, because this is chemotaxis we're talking about. So two kinds of kinases are involved, but what happens is that there is a chain reaction occurring here, if you can see. So this is more or less what we refer to as the phosphorylase or phosphorylase reaction, which we will see in a while, some other examples and a bit more explanation on it. And in B subphylase, there is also a TCS in the form of sporulation. So what happens in sporulation is that the reaction occurs that occurs in your histidine kinase is a bit different. The structure of histidine kinase is also quite different, because if you understood what sporulation means, which means that it will give out signal, but the signal is going to be divided. One signal is being given out from histidine kinase, but then it will be divided into two different response regulator to produce the expression. And more often, as you can see over here, only one of them will have the transmitter, or basically will give out the output, not both of them. Alright, so for the phosphotransfer system, histidine to aspartic acid is common, but for the phosphorylase system, as you have seen in the third example, it's basically in a sequence of reaction, histidine to aspartic acid, histidine to aspartic acid, in kind of like when you run in a baton, you pass a baton, so it's kind of like this kind of reaction. So this kind of modular organization of the TCS added basically they are added complexity. It also provide multiple regulatory checkpoint, and each point of integration between the signaling pathway provide another level of control of gene expression. So let's have a look at this classic case of the 2CS. So this is the plasma membrane. So your histidine kinase is situated there, and it is ready to receive the signal. So the input signal is here. So now we have different kinds of reaction over here, if you can guess what they are from the previous two slides. So we have autokinase, we have phosphotransferase, and we have phosphatase. So each and every one of them will basically transfer or autophosphorylate the signal, and then transfer it to the response regulator. Now for the autokinase over here, there is no response regulator, because usually this basically has to be phosphotransferate, or basically has to be autophosphorylated first. So what happens is that we have two case over here after the signal has been transferred or basically transmitted to the response regulator. So they will produce signals, for example either autoregulation or response gene. So that means that when you have autoregulation, which means that the signal will continue to be processed until it is ready to give a response. Or if it is sometimes really kind of like a gene that is needed for the survival, it will be autoregulated, which means that the expression is known and has been controlled and will be released or expressed, whether turn on or turn off automatically. Now the regulatory mechanism is as such that it actually involves a lot of the histidine kinase. Regulation starts at the histidine kinase, whereby we know that for the autokinase activity, it is either stimulated or repressed by specific stimulus. And for the response regulator, it will phosphatase, it is the phosphatase activity of the histidine kinase that will be modulated. For the response regulator, it is usually the phosphorylation of the cognate histidine kinase and dephosphorylation by specific phosphatases, and it depends on what kind of signal that it will produce, followed by stimulation of intrinsic autophosphatase activity. So apart from that, apart from these two, there is an inhibition of the phosphotransfer and also regulation of the expression of the two component proteins, the ones that will be the final product from the reaction. So a bit more on this, apart from basically providing another control or level of basically regulation for the genes, it is or the two component system could also be done via signal transduction, and this is done by transferring the phosphoryl group from the ATP to the specific histidine kinase in the basically by autophosphorylation reaction. What happens is that similar to the general reaction, so response regulator will phosphorylate the aspartic residue, and what we have is that the protein phosphatase for the histidine kinase. Once phosphorylation occurs, basically there is a conformational change in the response regulator, and this is done by activating the attached output domain. So the change in the response regulator, the conformational change or shape, will result in activation or repression of the target gene, turning on and off of the expression. And in this case, phosphorylation level controls the response regulator activity. So generally, or simply said, histidine kinase is actually bifunctional. It is able to catalyze both phosphorylation and also dephosphorylation of their cognate response regulator. Now, this is more or less a similar kind of diagram that you have seen previously, just that it is basically done in a different outlook. So what we have here is that it says that in this diagram it shows a regular blocking transcription, but activation can also occur. So this is where the gene, when the gene in the end or the output is that the expression is blocked, or repressed, but activation can also occur. As we can see that basically something is there. The response regulator basically block the RNA polymerase by saying that at the operator, so transcription is blocked. But you can actually activate this. How? That is one question for you. Now, in this next diagram, what you see is that this is a general two-component system, and this is for the chemoreceptor, for the chemotaxis, involving Ki A, which is quite common for a lot of bacteria. So, what happens is that take a look at the kind of response that it could give out. So, for example, using this both mechanism, so this one is for this, and this one is for this. But can you see that in the first output, this is actually a sporulation, because usually when you have the spore, it means that's for sporulation. So any kind of bacteria that sporulates, so they will of course produce this kind of reaction, which means that it is going to be in a relay reaction until the final product or the final gene expression, whether it is going to be activated or repressed, is produced. The second one is the classic case, whereby usually after the phosphorylation, basically one of the gene is basically stays there to receive the phosphotransfer, and then conveyed to the response regulator for the gene activation, another gene. And in the last one, so this is more or less similar to this, but as you can see, it is actually a part of the histidine kinase, not really standalone as the classic case or normal case of the two-component system, because basically it needs to be phosphorylated by the histidine kinase, not receive the phosphate group, then only to be dephosphorylated by the response regulator. So again, you can see a lot of these diagrams. So that's why I basically put different diagrams. Okay, you can basically get from over the internet, and all this comes from books. So more or less they have a different outlook, but the story is similar. Okay, so this is a chain reaction of phosphoryl. This is the classic case of the two-component system, and this is of course the most complex of all complex phosphorylate reaction, which we will see in a while. We're not going to go detail into this because actually one whole course could be held just for the phosphoryl reaction itself. Alright, so what we're going to take a look at in the next 3-4 slides is actually on the how the signal is being integrated. So for example, the first one, so like I said when you see the SPOO genes, you will know that it is actually for the sporulation. So over here an example, 5 related histidine kinase are capable of phosphorylating the response regulator spoof. So all of the kinases share sequence similarities surrounding the phosphoryl table histidine residue, but differ in their sensing domains, of course. So what happens is that there is another gene here or protein is expressed during the vegetative growth, and two other gene RAP-A and RAP-B are induced by the COM-A and also the COM-P TCS. This is actually the component system, two-component system. So sporulation in this case is prevented during the vegetative growth, and you get competence development. Now in certain cases sporulation is needed for the growth for population or overpopulation of certain bacteria, but in certain cases when the cell density is too much they need to control it or prevent it. So this is actually one of the mechanism for integration of signal using two-component system to halt or to control the overpopulation. Second example of signal number 2 is basically a chain reaction. So over here we have RAS E and RAS D which is the basically another one component system which regulate expression of genes required for anaerobic respiration, and we have another two TCS which regulate expression of genes required for phosphate uptake. So when the phosphate is low, so phosphorylated 4 basically what happens is that over here will induce the expression of the RAS operon while repressing the 4P independent promoter. So what happens is that you will not get anything transcribed. So phosphorylated RAS-D activate the 4P and 4R expression to give a positive feedback loop. So which means that even though they couldn't activate this but it basically jump to another two TCS to activate another gene needed to compensate for the low phosphate. In the third kind of signals or integration of signal, so we basically have 3 two-component system, and the list could go on and on and on. So but don't worry, I'm only giving you up until 3 plus another one which is on the phosphorylate. So for this case the product of the UDG which is over here, the final product, see is required for both the PMR regulated modified so from here to here. So what happens is that both the PMRA and also PMRD can bind and activate transcription of the UGD promoter, and the PMRD the ones that is made when there is magnesium is being activated post-transcriptionally and independently of the PMRB in response to magnesium. But the UGD gene is only expressed in response to magnesium, ferrum and also or for the cell envelope stress when it occurs. So that means that there are 3 kinds of mechanism to get the UGD to be expressed and there are 3 different TCS that could be used to transcribe or to control the UGD expression. So very complex, right? And this is basically how bacteria control certain gene, how it should be expressed, how it should be repressed. Now the final one, and I think this is the last slide, on phosphorylae. Okay, and phosphorylae is usually but not strictly, okay, available, okay, for virulence gene control. So what happens that, okay, for example, okay, over here this is basically in B. pertussis, Rottenella. Okay, so what happens, okay, is that when a gene, okay, or a system or bacteria, okay, is basically, okay, has a virulence gene, what will happen, okay, to the bacteria in general? So sometimes, okay, what will happen, okay, is that it could destroy the bacteria itself. It could overpopulate, okay, and basically it could kill off, okay, one another in the population. But okay, it could be controlled via phosphorylae. Okay, so this is how it's done. Again, like I was telling you earlier on in the phosphorylae, in the 4 sequence slide. Okay, whereby, okay, we have, okay, a kind of chain reaction occurring. Okay, the phosphatase, okay, a phosphate group, okay, is actually being passed, okay, between histidine to aspartic acid to histidine to aspartic acid until, okay, it reach, okay, the final gene, okay, to be expressed. Okay? And for example, okay, in this case, in B. pertussis, okay, there are 4 class factors, okay, that basically could be produced as a result of the phosphorylase reaction. Okay, you could either get, okay, these 2 genes, okay, being expressed, okay, or these 2 genes, or this 1 gene, and okay, the last one would be the virulence repress the genes VRG. Okay, this is the most virulent gene which will cause death. Alright, so all in all, okay, this is a bit, okay, or the summary or what I could basically deliver on 2 component system, okay, as only to be taught in 3 hours, okay, plus discussion and whatnot. Because as I was saying earlier on, okay, TCS is actually a very complex mechanism, and for each bacteria, how they use it and how they control certain gene using TCS is actually very complex, and a lot of them are poorly understood. Okay, and those that are understood, okay, from the publication paper, basically solely focus, okay, on how the 2 TCS work, okay, in that system alone, or for that gene production alone. So if you're interested, go and read, okay, some papers. And of course, okay, for this TCS, we have an activity for you to help to help you understand the TCS better, of course, by reading journals and by working in group.